Okay, good afternoon, everybody. I'm very sorry that I've had to do these classes this week as a recorded asynchronous class, but I really don't want to be spreading virus everywhere, even though this is a microbiology class. I don't have COVID, but I do have a cold. But in these times, please remember, no matter what you think you may have, please do not come to the college and spread it around, because other people then have to deal with the decision about what it is that they actually have. It's as simple as that. Also, forgive me if my voice kind of gives out halfway through this class. Okay, so what I'll tell you is my name is Dave Muir, and I'm going to be teaching you this semester. I am going to jump straight into the class material today and tomorrow. And then when I meet with you probably on Monday for the first time, I will go over the class regulations and everything else. But for the moment, let's just stick to our class material. These lectures are, the PowerPoint presentations are all uploaded to Canvas for you. Please make sure that you refer to them and that you print them out using the notes facility, so that you save paper. But I strongly advise you to print these out and use them to make notes during the class, whether you're sitting looking at it online, as you will be this week, or whether you are in class with me. Okay, so this is microbiology 300, and it's actually the second semester that we offer microbiology. And this course in microbiology is intended more for people who are doing biological degrees rather than for people who are doing health sciences. Although if you're doing health sciences, that's absolutely fine. It's just that there is, in this semester, we put a little bit more emphasis on things like genetic engineering, environmental microbiology, those sorts of things, rather than on clinical microbiology, as we do when we teach for the health sciences. Okay, so let's just have a think for a moment about how easy it is for us to conceive of and even to visualize the presence of bacteria, things like that, in our environment. We are constantly told about these things. We're constantly warned about them. And when we fall ill, very often we are diagnosed and we are told, you have this bacterium, you have this virus, and we accept that that is the case, even though maybe we've never seen these things or whatever. Just think for a minute how much work it has taken and how far humankind has had to develop in order to arrive at those simple conclusions. It was not easy, because these things, for example, are all essentially invisible to us. The symptoms of disease, of this sort of thing, some of the properties of these microorganisms are visible to us by the processes that they engender, but they themselves are essentially invisible because they are simply too small to be seen. The ability of the human eye to resolve an object is about 0.1 of a millimeter. Anything smaller than 0.1 of a millimeter, our eyes simply are not structured. Our lens structures, our retinas, our corneas are not structured to see anything that small. And even a small object, 0.1 millimeter across, we can barely see, and we discern it as essentially as a point, because we're unable to resolve the two sides. Anything smaller than that is essentially invisible. It has been, for much of mankind's history, there was a huge debate about what it was that caused disease, for example, about what it was that constituted life, living things. What was it that made something alive and another thing dead, etc., etc. And it was the resolution of this dilemma that things were too small to be seen and yet could still exist that began to resolve some of those controversies here for human beings. What I mean by that is there was a school of thought that said if you couldn't see it, it didn't exist. There was another school of thought which said that in fact, life existed on a much smaller scale than what we could easily see. Now, a lot of this early information came from the fact that wealthy gentlemen, particularly in the 16th, 17th century, began to take up natural history as sort of a hobby. When you were wealthy, you collected things. You, as a hobby, studied things. You maybe collected flowers. You maybe collected butterflies, whatever. A certain subset of these gentlemen became extremely interested in the use of lenses. Now, lenses for eyeglasses, for magnifying, ordinary magnification, had been known since Roman times before. But around about the 17th century, they began to develop very skillful ways of manufacturing lenses and began to examine the world around them using these lenses and amplifying now to such an extent that they began to just be able to see objects which were otherwise too small for the human eye to see. Now, this wasn't particularly in terms of our modern day ability. This wasn't particularly spectacular, but for them it really was, because a whole invisible world began to open up and become visible. This gentleman, Robert Hooke, in 1665, made a very important discovery. He actually looked initially at wine cork. Now, the corks that you use in wine bottles are from the bark of a tree, the cork oak. So we know that that is basically cellular in structure. It's manufactured by the cells of the oak tree. They knew nothing about cells. They didn't know anything about it. But when Robert Hooke examined thin slices of cork using a good lens, he could see that the cork was composed of little tiny boxes. Now, this was just the remains of the cell walls of those cells. They were not living cells, but he went on to look at many other things. He looked, for example, at fungi with a good lens and saw that they too were composed of what looked like these boxes or these little compartments, which he was the first to call cells. And we know now that the cell is the basis of life. The cell is the single characteristic that characterizes all living things. And at its simplest, the cell is defined as cytoplasm contained within a membrane, which allows the interior of the cytoplasm to be different to the exterior of the cell. And contained within the cell is hereditary material. The early workers knew nothing about that. So this is the beginning of cell theory. Robert Hooke, seeing these cells, he began to make the assumption that all living things, in fact, were composed of these little building blocks of the cell. Now, one of the most important strides was made by this gentleman here. This is Anton van Leeuwenhoek, who is a Dutchman. And he was a merchant, quite a wealthy chap, who was absolutely obsessed with manufacturing lenses. And he had the ability to make a very, very small, almost spherical lens with a very high magnification. And the problem with this lens of his was it's very difficult to use because it was almost spherical, the focal point And the image it made lay very close to the lens itself. Very difficult to use. Couldn't hold this little tiny lens and look through it. He manufactured a little machine to use it. And this is it here. It consisted of a plate like this. Here is the lens there. He would manufacture these small lenses and embed them in a hole in this plate. So he could hold this plate like this in front of his eye. His great innovation was that he made this little point here on which he could place a droplet or a specimen. And he could actually, here there was a screw which allowed him to move that back and forth and back and forth. So he could hold the plate in front of his face like this and move the screw back and forth and have it in bring it into perfect focus. And using this very, very simple machine, which is the very first true microscope, he was able to begin making some absolutely stunning observations. Now, first of all, Van Leeuwenhoek was an innovator, first of all, because he was an inventor. He wasn't the only person looking at things with high-powered lenses, but he did develop a very good, simple microscope. The second thing that was important about him was his insatiable curiosity. He looked at absolutely everything that he could possibly lay hands on. He looked at blood. He looked at pond water. He had diarrhea and examined his own diarrheal stool. He looked at his own semen. He looked at pollen. He looked at all sorts of things. And that was the first thing, his insatiable curiosity. And then thirdly, the fact that he wrote everything down. He made careful notes and drawings of everything that he did, and he communicated that with other people. In particular, he communicated with the Royal Society in London. He also communicated with other gentlemen in the Netherlands. They would get together and talk about their findings, and they would spur one another on. And they began themselves to follow Van Leeuwenhoek's lead in making these little simple microscopes and beginning to use them to examine the world around them. Van Leeuwenhoek's discoveries were monumental. First of all, here are some of his drawings of things that he observed. And you'll see, first of all, he was the first person to observe coccoid bacteria. And here they are here, carefully drawn out. This is probably Staphylococcus that he's observing here. Little coccoid bacteria, they are spherical, very small, and often they will pack themselves together like this. He was the first to observe rod-shaped bacteria or bacilli. And here they are here, some larger than others. He was the very first person to observe and document that these bacilli actually moved through the medium. They moved through water. He saw many, many other organisms. He saw many protists, for example, eukaryotic organisms in water. He even saw disease-causing organisms. For example, he was the very first person to identify and draw an organism called Giardia, which is a protozoan which causes diarrhea. He himself had diarrhea caused by Giardia. It was very, very common. He actually saw and drew that organism, which is, I have to tell you, a monumental feat, because even with modern day microscopes, it's quite difficult to see and to draw. So here comes now from a state of humanity where we didn't believe that anything existed if we couldn't see it, all of a sudden explodes out this world where, in fact, it is shown that these microorganisms are present all around us. Van Leeuwenhoek showed that they came from the skin, from the saliva, from the teeth, from the soil, from all over the place. People like Robert Hooke came and said, we ourselves are composed of these small units, or the cells. And this is a revolution in the thinking of humankind. Not only that, but it began to engender this debate about what constituted life itself now, and where these organisms that they were seeing came from. Van Leeuwenhoek called them animalcules. Anything that he saw that was revealed by his microscope, he called it an animal, a little animal. There was no clarity about what they actually were, nor about where they came from. Now, for a very, very, very long time, since thousands of years before, there had been this theory that life arose continuously, that life could be generated under many circumstances. That if you took a broth, you took soup, for example, and you left the soup, after a while, it would begin bubbling and fermenting and growing stuff. And even when the early microscopists examined it and saw little animalcules, it was believed by many people that those had arisen spontaneously, that the fluids themselves in decaying had generated these organisms. And this was called spontaneous generation. And it arose from the idea that the earth is saturated with what is called a vital force, a force of life, which finds its expression wherever it can. It forces itself into being. And this manifests itself as life arising spontaneously. So, for example, ludicrous as it sounds to us, they believed that rotten meat generated maggots. Some people believed that mud, pond mud, generated swallows. All of these firmly held beliefs contributed to this totally erroneous idea that life is generated continuously. And this sat like a dead weight on the study of biology for centuries, because if you did an experiment, every result could be confounded by this notion that life arose spontaneously and did things independent of whatever you had conditions you had set up. The counter argument to that was the belief in biogenesis, the idea that in fact all living things arise from other living things. There was an original event, an original creation of a living cell, and that in fact all living things have been derived from other living things, that we no longer spontaneously generate life, but that all cells arise from pre-existing living cells. And Van Leeuwenhoek believed this very strongly. He did not believe that the animalcules that he saw were generated spontaneously. He believed that they existed in these environments and that they arose from one another. And for example, he took this argument about rotten meat and said that maggots arise from eggs which are laid on the meat. And he knew that because he found the eggs and he examined the eggs and saw that they had small developing embryos inside the eggs, which gave rise to the maggots. This was still not good enough for the spontaneous generation people. One man called Francesco Redi, an Italian, did a simple experiment. He took meat and he put some meat in an open jar, and he put some meat in a jar which he covered with gauze. And in the open jar, the meat rotted, and became covered with maggots. In the jar which was covered, the meat rotted, but it did not become covered with maggots. And he said, as a conclusion from this, that this was because in order for the maggots to be produced, adult flies had to have access. It's hard for us to believe that anybody would argue about this, but it is true that they did. Human beings, once they have an idea in their heads, will twist reality and fact to fit what they believe. A scientist does not do that. A scientist tests reality and draws conclusions based on this. And this is why the simple experiment of Francesco Redi's is so important in biology, because it was a true scientific experiment. What he did was he set up contrasting conditions. In the one condition was the normal condition, meat exposed to the air and obviously to flies. The other, so that is a control. That is a positive control. This is his experiment. He's taken a jar, and he does not know, he's effectively pretending he does not know the fate of the meat that is in these jars, right? He's taken this jar and he's covered it with gauze. This is his experiment. He's manipulated this normal situation to see what the effect is. And what he sees is that in this jar here, there are no maggots. In this jar here, there are maggots. Not only that, he had another little trick up his sleeve, and that was he examined this gauze and he saw eggs laid on it. And he drew the conclusion, correct conclusion, which van Leeuwenhoek said: flies lay eggs. Maggots arise from already existing flies. In order for the maggots to arise on meat, the fly has to have access. So that seems to us perfectly obvious, right? But the spontaneous generation people were still not satisfied. And they said, well, look at this meat rotting in the jar. The meat is still rotting. After a while, the meat begins to liquefy, it begins to grow things, it begins to bubble, begins to look like it has other living things. They say, the spontaneous generation people now say, although the flies can't get through, spontaneous generation can still go on because the vital force has not been prevented from entering the jar. I know these arguments sound ludicrous, but they went on and on for decades, arguing back and forth about whether spontaneous generation occurred or not, despite the clear evidence in front of them. In 1745, this gentleman, John Needham, performed a similar experiment to Redi's, excepting that he used this broth idea. And he decided that what he was going to do was he would take broth and a soup, essentially, and he would boil it, and then he would leave it and see what happened. Well, if you do that, after a while, the broth begins to ferment. And if you examine that fermentation, you'll see that there are animalcules swimming around in it. Now the question gets asked, where do these animalcules come from? The spontaneous generation people say that they come from the vital force generating life. The biogeneticists say no, they've come from somewhere else. Needham was a biogeneticist. So here's his experiment. It is elaborated on now by this gentleman, Lazzaro Spallanzani, who elaborated on Needham's experiment, because what he did was, here's Needham's experiment: boil the broth, leave it open, and after a while you get growth. Now Spallanzani takes the broth, boils it, and he seals the flask, and nothing happens. There's no fermentation, there's no growth of anything, there's no growth of animalcules in the flask. And then take the top off and growth begins. the spontaneous generation. People say, there you are, you see, you've proved it. What you've done is you've sealed off the vital force. So nothing can grow because the vital force isn't. But as soon as you allow the vital force access, then these animalcules spontaneously generate. Can you see what I meant when I said every experiment you do gets confounded by the spontaneous generation, people? This is why this idea was so important, was because it steered science in completely the wrong direction for centuries. Here's the very first formulation of the idea that every cell arises from a pre-existing cell. And it's by this man, Virchow. And Virchow proposed that every cell arises from a pre-existing cell, that there must have been an original founding event where a cell evolved. But once that cell successfully evolved, all other cells are derived from that cell, from that original cell. We still believe that that is true. The whole idea of spontaneous generation, this was only laid to rest in 1860s. And that's less than 200 years ago. And it was laid to rest by this very famous person, Louis Pasteur. And Louis Pasteur can probably be designated as really the world's first true microbiologist. By this time, by 1860s, they had developed compound microscopes, such as you use in the lab. So their ability to visualize things was very, very much better. But this idea of spontaneous generation was still around. And so Louis Pasteur thought it was absolutely ludicrous, and he designed these experiments. He said, okay, I'm going to satisfy all of the conditions for the spontaneous generation people say we have to have. I'm going to do the broth experiment. So here it is here. He takes a flask and he makes a broth, and he then boils that broth and kills any organisms which might be in it, kills the animalcules in it. He now got to set this up so that there is the vital force has access to the broth. So what he did was he made these flasks with a long neck, and he bent the neck like this into an S shape. It's called a swan-necked flask. Here's his broth. He boils the broth vigorously, allows all the steam to escape, and now he just cools it. And he lets it cool, and lo and behold, no microorganisms grow. This broth here remains sterile. And in actual fact, if you want to, sometime, if you're in Paris, you can go to the Pasteur Institute, where there are still flasks which contain broth, which Pasteur boiled in swan neck flasks, which is still there, and it is still sterile. They have since sealed off the flask because they don't want the broth to evaporate. But the point being that here, Pasteur has done everything. He's taken a broth, he's boiled it just as everybody else did, but he hasn't sealed the flask. He's left the flask open to the vital force if it exists, and yet it still does not generate life. Spontaneous generation does not occur. If, on the other hand, he takes this flask and he tips it over so that some of the broth contacts the neck, and then he tips it back up again, then the broth becomes contaminated. Here's the reason why. This part here is a trap. It's open to the air, but the thing is that bacteria, microorganisms in the air do not fly around. They move as particles themselves, or they move on particles. They largely travel on particles of dust, skin flakes, things like this. Those things, and the bacteria themselves, in actual fact, they have weight. And if you put them in absolutely still air, they will settle out. In order to get bacteria from here to here, you would have to have a draft that blew them in. And that does not occur in this flask, in this structure. Instead, any bacteria that settled out from the air will settle here. They won't make their way all the way through here because there's no mechanism for them to get there. Only if you provide them away by tipping the flask and then allowing it to swill back again, only then does your broth become contaminated. It was the nail in the coffin. That was the nail in the coffin for the spontaneous generation theory. And Pasteur was the first to conclusively demonstrate all cells arise from other cells. They do not arise spontaneously. The second thing that this experiment of Pasteur's demonstrated, and all of his experiments showed, was that these microorganisms occur everywhere. They are in the air, they are in the water, they are on our skins, they are all over the place. You can set up conditions where you exclude them, but essentially that has to be a very special circumstance because they are in fact all over the place. They are ubiquitous. The second thing that Pasteur has conclusively shown, despite the fact that they are invisible, microorganisms are actually alive. And provided with the right conditions, microorganisms will reproduce. That is why these broths become contaminated. Once you get a few microorganisms in them, those microorganisms reproduce very rapidly and end up contaminated. These are all revelations to scientists at the time. Many of them arose from this one worker, from this one laboratory, and that is the laboratory of Louis Pasteur. So these gave rise to an explosion of interest in the study of microbiology, of those organisms too small for us to see easily with the naked eye, but which were capable of doing all sorts of different things. And now scientists set about trying to work out what were these organisms and what were the things which they could do. In particular, they became interested in the involvement of microorganisms and disease. And not only disease of human beings, disease of animals and disease of plants as well. They also began to be interested, largely because of the work of Pasteur again, in the interaction of living organisms such as human beings and disease-causing organisms, in particular, how we fought off those diseases. And that led to the study of immunity, of how the body reacts to the presence of these foreign agents. And lastly, it led gradually, it has to be said, but it ultimately led to study of antimicrobial drugs, of compounds which we could take, which could be directed at disease-causing organisms. Now, first of all, I should tell you that there are many, many people who refuse to believe this at all. And they particularly refuse to believe what Pasteur referred to as the germ theory, that microorganisms could actually cause disease. It seemed to many medical people ludicrous that what they called these little plants that flew around in the air, as they visualized it, could end up causing diseases which were so severe that they could actually impact human population, whole human populations. It was inconceivable to them that something invisible, for example, could have caused the Great Plague, which had killed more than a third of Europe's population in the 1200s. Nonetheless, Pasteur and allied workers in the golden age of microbiology set about showing that that was exactly what microorganisms could do. First of all, microorganisms do not arise spontaneously from non-living matter. They are generated by other microorganisms. Microorganisms are ubiquitous, and microorganisms are diverse. There are many, many, many kinds, and they do different things. Some of them cause disease, some of them are useful, some of them are involved in processes in biogeochemistry, all sorts of things. They are incredibly diverse. They do many different things. And this is what the early scientists now set about doing. So one of Pasteur's most important pieces of work was an early piece of work which he was conscripted to do, which was to work with wine farmers and to examine the production of wine from fermentation processes. Fermentation itself was a controversial topic. Many people believed that what was happening in fermentation, when you took, for example, you took grape juice and you left it alone, it began to ferment, bubble, produce alcohol, etc., etc. Many people believed that that was simply a breakdown, a spontaneous breaking down of the compounds in the wine, in the grape juice. But Pasteur did not. Pasteur examined these solutions which were fermenting and saw that there were microorganisms present. They were not bacteria, they were yeast. Nonetheless, they were microorganisms. And he became involved in two aspects. First of all, the wine farmers had tremendous problems because periodically a batch of wine would become infected with bacteria and would turn to vinegar or be spoiled in one or other way. Second of all, they knew that different kinds of grapes and different combinations of conditions produce different flavors, and they were interested to know whether this was controllable. So Pasteur, first of all, showed that the spoilage of wine, it turning to vinegar, was the responsibility of a specific group of bacteria. And he second of all showed that if you took grape juice, effectively, that was you wanted to turn to wine, and you heated it up, not to a very high temperature. This is not sterilizing the way you would if you were to boil something. This is just heating it up to about 65 degrees Celsius. If you did that and kept it there for some minutes and then allowed it to cool and protected it from recontamination, you could kill off in the wine juice, you could kill off the bacteria which cause spoilage and leave alive those which were the useful fermenters. So this was its process called pasteurization. And pasteurization was a revolution in public health medicine because pasteurization could be used on many different fluids. In particular, it could be used on milk. And milk at that time was a very dangerous product because it quite frequently contained bacteria which were harmful to human beings, such as tuberculosis, bovine tuberculosis bacteria, and various other things as well. Milk could be pasteurized, and potentially pathogenic organisms could be eliminated from it while minimally affecting its other properties, hardly changing its flavor, for example. In the case of wine, not affecting the process of wine formation. So the other thing that Pasteur did was also extremely interesting. He began to isolate different kinds of yeasts and purify them, and found that he could tell farmers, you use this particular yeast and you get these particular properties. He began to pedigree the microorganisms. What he was doing in doing that was demonstrating that particular microorganisms have particular properties, that if you are able to distinguish one microorganism from another, you can distinguish it by what it does, by its observed properties. Okay, so there's one little caveat which I have to add here. Okay, I just want to go back here to Pasteur's experiment using broth. Pasteur was very, very lucky. He was extremely lucky. He was very lucky that when he did this boiling of his solutions, he only had bacteria in here which were busy reproducing. Those we call vegetative bacteria. They are active, they are growing, they're reproducing, dividing, etc., etc. They're quite easy to kill by boiling. But he was lucky he did not have bacterial spores in there, because bacterial spores are a special cell type, which we'll learn about later, which have virtually no metabolism and which are resistant to boiling, but they will grow, they will germinate once conditions are suitable. So these experiments would have failed if he had had spores in there. And he was just lucky. This set of experiments, he didn't have any bacterial spores. He was only dealing with vegetative cells. Spores are a tremendous problem in public health and all sorts of things, because bacterial spores are resistant to boiling. They survive at 100 degrees Celsius, and they also are resistant to most disinfectants and things like that. Things like alcohol and that sort of thing have very little effect on bacterial spores. This had to wait for more microbiological work for them to realize. Okay, so Pasteur's ideas were naturally very easily transferable to the realm of health. People began to think, look, maybe microorganisms are actually involved in disease. And if we can eliminate microorganisms from an area, maybe we can prevent people from actually acquiring disease. And this was positively demonstrated by this gentleman, Joseph Lister, followed Pasteur's work very, very closely. And Joseph Lister was a surgeon. Around about the same time, the whole of human surgery underwent a major revolution, because these two things happened. First of all, they developed anesthetic. The first anesthetics that they used were things like chloroform, which could actually render people unconscious during surgery. Before that, surgery had to be done with a patient fully conscious. Sometimes they would get them blind drunk on alcohol, maybe fill them up with morphine if they had it available. But essentially, surgery, that you held people down and did the surgery as quickly as possible and hoped that people would survive. But surgeons at the time were the renegades of the medical establishment, and they paid no attention whatsoever to cleanliness. In fact, most surgeons took it as a point of pride that they would wear their apron. They'd wear a leather apron, and the filthier that the apron was, the more skilled you assumed your surgeon was, because it meant that the surgeon had done so many operations. So you can imagine that surgeons themselves were a source of major contamination during surgery. Now, Lister reformed that whole idea. First of all, he had access to anesthetic to anesthetize his patient. But second of all, he, as far as possible, sterilized his surgical environment. He used carbolic acid, which was a known disinfectant, and he would spray the whole room, the whole area. Here they are here. This is an actual photograph of Lister at work. They would spray the whole area with this mist of carbolic acid, which is phenol, very, very, very caustic and very unpleasant to work in those environments. So they still worked quickly. But they also developed methods for example, sterilizing the wound, sterilizing the surface, the skin surface before cutting somebody open. and operating as far as possible under conditions which eliminated the likelihood that there would be a contamination. He was also a proponent, simple as it is, of personal hygiene, of washing hands, etc., etc., when performing surgery. And he positively showed that his contamination rates, his death rates, were much, much, much lower than any of the other surgeons. The other surgeons, by the way, laughed at this. They thought it was absolutely ludicrous way to behave. But of course, patients heard that if you went to Lister, you survived. If you went to the other surgeons, you died. So Lister became a popular surgeon. So a lot of Lister's ideas were based on work which had been done, poorly advertised, but nonetheless known by this gentleman called Ignaz Semmelweis. Now, he was an Austrian doctor, and he worked primarily in obstetrics wards. One of the most dangerous things that a woman could do at this time was to give birth. But even more dangerous was to give birth in a hospital environment, because there was a very, very high rate of what was referred to as puerperal fever, a fever of childbirth. And fever of childbirth was actually due to a bacterium, to a streptococcus, and called Streptococcus pyogenes, which could infect a woman's reproductive organs, first of all, the vagina, cervix, and then the uterus, and then spread to the rest of the body and cause a fatal sepsis. And women very often died within days of giving birth because of this puerperal sepsis. So what Semmelweis did was to examine the rates of puerperal sepsis. Now, here's an interesting comparison. You see this blue line here? This here is a hospital in Dublin. Now, this is an ordinary hospital, right? And you can see here the rates of puerperal sepsis. It's definitely not zero, okay? It's prevalent, it's still not prevalent, but it's still occurring regularly inside this hospital. That's point number one. Here's the hospital where Ignaz Semmelweis worked. These are their rates of puerperal sepsis. You see this line here. Up until that line, the rates of sepsis in the Austrian hospital were much the same as those in the Dublin hospital. Now, what made the difference here? The difference there was that this hospital began doing autopsies. It began doing autopsies on women who had died of puerperal sepsis. This had not been done before, but the autopsies were done by the same surgeons who were then going and delivering babies in the obstetrics wards. So they were cutting open dead bodies of women who had died of streptococcal infection. They were getting streptococcus all over themselves, on their hands, whatever. And they were then going and delivering children, delivering babies in the rest of the wards. And it was no wonder that the rates just took off. This is where Semmelweis arrived. He arrived at this hospital and saw the incredible rate of puerperal sepsis. And there was a single event which convinced him what needed to be done. And that was, he had a friend who worked there. He was a surgeon doing the normal surgeon thing. And this surgeon pricked his finger during an autopsy of a woman with puerperal fever, who had died of puerperal fever. And this same surgeon died. He died exactly the same symptoms as somebody who had puerperal fever. And Semmelweis made this intellectual leap that somehow an infectious particle had been donated to this friend of his, and he had then subsequently died. He then instituted a system where he required everybody entering his wards, nurses, doctors, everybody, to wash their hands with a disinfectant solution. It was essentially bleach. It was a chlorine solution. Here's where that began to happen. There he is there. And look at the rates of puerperal sepsis. They plummet all the way back down to this background level. It's not zero. There are many other things going on. They're not sterilizing bed linens. They're crowding people together in wards. All sorts of other bad things are happening. But this simple measure, washing the hands and sterilizing the hands alone, stopped this direct transmission of streptococcus from surgeon to women who were delivering. Unfortunately, Semmelweis was a very difficult character, and he didn't get on well with any, and instead of like trying to persuade people, he became very, very autocratic about this, and he ended up being booted out of this hospital, and eventually, unfortunately, became so bitter about the whole thing, he actually became mentally ill. But he positively demonstrated that a simple measure like washing your hands could have this major effect on transmission rates of a pathogen. And it was those kinds of ideas which engendered Lister and all of the rest to go into a system where they behaved as if these organisms were present, even though they could not see them. Other health workers thought this was ludicrous. It was a ludicrous idea that something that you couldn't see could cause these diseases, but they said no. The work of Pasteur, the work of Semmelweis show us that these things are present. They are present everywhere, and that if you limit their access to the human body, then you limit the rates of illness which they can cause. So this is referred to as the germ theory of disease. And this was taken up by another very, very famous microbiologist called Robert Koch, who was a German. He was an arch rival of Pasteur. They did not get on well at all, even though they fed off one another's work the whole time. And Koch worked specifically on one organism called Bacillus anthracus. Bacillus anthracus is a rod-shaped bacterium which causes the disease anthrax. At that time, anthrax was a very common disease. It was most common in domestic cattle and sheep, and caused mass mortalities, and could be transmitted to human beings, where it could also be fatal. But it was very important economically because of its impact on agriculture. And it produced very specific symptoms in the animals. It was easily identifiable that this animal had the disease of anthrax. So that's an important point, because Koch wanted to set about showing that there were ways to determine the particular disease that a particular organism could cause. He set about drawing up the rules whereby you could tie a disease to an organism. And these are known as Koch's postulates. Then they demonstrate a specific microbe causes a specific disease. So this is what he did. He worked on Bacillus anthracus, and here it is here. Easily recognizable. It's a spore-forming bacterium, which is one of the really important things about it. It's an anaerobic bacterium under many circumstances, but it can survive for a very, very long time in soil and in water, but especially in soil, as a spore. And you can see it there. There's that clear area there, that is a spore. The rest of it, that's the vegetative cell that's growing. So his first postulate, he wants to show that the same organism is present in every case of the disease. That's postulate number one. Whenever he sees these symptoms and he goes and he takes samples, he sees that he gets a similar organism. Second of all, when he gets this organism, postulate number two, you must be able to culture that organism. So this can be done. This he did quite easily with Bacillus anthracus. He could grow it in culture. And when you grew it in culture and examined it, that was the only organism that was present in the culture. That is called a pure culture. So postulate number two, you can extract this bacterium. You can establish pure culture. If you take that pure culture and you inject it into an uninfected experimental animal, the animal acquires that disease, that specific disease, shows those specific symptoms, and in this case, anthrax, it dies. Postulate number four, probably the most important one of all, and that is when this experimental animal dies, you are able to extract exactly the same organism from it as that you grew in culture and as that you had originally extracted from the original sample. So what you have done is you've tracked this organism through from the natural state, the disease, through to an artificial state, the culture, then to an artificial creation of the disease again. You've reproduced the disease, and you have then been able to re-extract the organism, the same organism again. It has to be the same organism all the way through. And if that is the case, then you can positively state this organism causes this disease. This organism is the cause of this disease. This was an extremely profound finding because it opened up the possibility for people to examine diseases of many different kinds. Obviously, not all diseases are caused by microorganisms, but many of them are. And it opened up the possibility now that for many diseases, such as tuberculosis, for example, you could actually go find microorganisms, track them, culture them, and show that they could cause the disease. And that the great hope then was this: once you know that, once you can identify the disease-causing organism, maybe you can work on that organism more, find ways of limiting its disease-causing capabilities, either by public health measures, for example, pasteurization or whatever, or ultimately by developing drugs, by developing ways of trying to hit at this particular organism and stop it from growing and being able to cause disease. Koch was really important. We often refer to him as the first bacteriologist because he worked specifically on bacteria, and especially those which cause disease. And he was responsible for showing that there's many different ways of culturing bacteria to pure culture. So he developed pure culture techniques, which enabled people to isolate particular kinds of bacteria and then look at their properties. So the one little side story that I want to tell you, we're talking here about Pasteur's French and Koch, who is German. All these Europeans were the leaders of the golden age, but there is an American tucked away inside the story. And that's this lady here. Her name is Fanny Hess. And her husband was actually working for Robert Koch in his lab. He was also an early microbiologist. So one of the important steps, experimental steps in developing pure culture, is to be able to streak out bacteria onto solid medium. When you look at a liquid medium, at a broth, it grows bacteria fabulously well. But when you just look at the broth, you can't tell what is actually growing there, because you can't see the microorganisms. If you have a solid surface, then you can actually spread out bacteria on there. You can spread them out to such an extent that you separate individual bacterial cells one from the other. And each bacterial cell will grow and will form a colony. And those colonies very often have recognizable characteristics. And you can sample from the colonies, and then you can then examine them under the microscope. You can put them through different experimental biochemical experiments and things to characterize them. But you can get reproducible colonies by streaking them out on solid media. So solid media are tremendously important. Koch initially developed solid media by adding jello, to his media. So a solid medium is just a liquid medium, a liquid nutrient broth, for example, that you add a solidifying material to. He added gelatin initially. The problem with gelatin is that many bacteria love gelatin, and they produce enzymes which break it down extremely rapidly. So it's very difficult to grow them on a medium which is solidified with gelatin. But this lady, Fanny Hess, the wife of somebody working in his lab, she used to make her own jellies and jams in her kitchen. And to solidify her jellies and jams, she used a substance called agar. Now, agar is extracted from seaweed. Very few bacteria, apart from marine bacteria, ever see agar. So they don't have the enzymes to break down agar. And she showed this to her husband. And he took agar into the laboratory and used agar to solidify the mechanisms, to solidify the medium, sorry. And it worked fabulously well. And the bacteria didn't break down the agar. The surface stayed solid. And ever since then, we've used agar as the principal solidifying medium when we make up bacteriological media. And that's all thanks to this lady, an American called Fanny Hess. Okay, so I think that I'm going to stop there for the moment. I'm losing my voice for a start, but we'll proceed from this slide on for the next time. And I repeat, I am actually going to do a recorded class on Friday as well, just to be absolutely on the safe side. We'll have a recorded class on Friday as well. And I will see you in person on Monday. And on Monday, we will go through the class rules and grading and everything else. Okay, so I'll see you then.